Hi, my name is Mariah Snelson. I am a junior biomedical engineering major researching the effects of aging on cell motility in a 3D microenvironment in the Philip Lab under my mentor, Anshika Agrawal. Aging is a physiological process that is characterized by the accumulation of cellular damage that increases risk of disease. Research to identify biomarkers of aging is key to mapping aging trajectories and predicting and preventing the onset of age-related disease. These biomarkers and their properties are encoded within the biophysical properties of cells. Of particular interest is how cell motility changes with age. Previous studies in a 2D microenvironment have shown that cell motility tends to decrease with increasing age, not because all cells become less mobile, but because of the enrichment of cells expressing lower motility and depletion of those expressing greater motility. Investigating cell motility changes in a 3D microenvironment will provide a more physiologically relevant ex vivo model to understand the association between aging and cell movements. We hypothesize that the observed decrease in cell motility in aged cell samples is not due to a population-wide decrease in cell motility, but rather from a redistribution of cells among distinct cell motility patterns within 3D microenvironments. Currently, most cell motility studies rely on bulk average patterns over time. The Philip Lab has previously shown that studying cell motility at the single cell level is more capable of accurately predicting cellular age in primary dermal fibroblasts. Additionally, previous studies have shown age-dependent redistribution of 2D cell motility states, along with a decrease in cellular heterogeneity with age. In this study, we aim to classify the heterogeneity of 3D cell motility states during aging by looking at the distribution of motility states and their age-dependent changes. Understanding 3D cell motility is critical to identifying the migratory properties of cells, which can provide robust information regarding cancer metastases, immunological responses, and differing aging trajectories. For experimentation, de-identified human dermal fibroblasts taken from donors of various ages are cultured and plated in type 1 collagen gels. Cells are seeded at a low density, incubated for 48 hours to allow for adherence and growth, and imaged every 5 minutes over the course of 8 hours to observe cell movement over time. Programs are used to track cell movements based on XYZ coordinates and time. The data is analyzed to identify patterns of differences between younger cells and older cells on a single cell level. In order to perform this analysis, the 3D microenvironment in which the cells were grown needed to be optimized. The results of our experimentation this semester indicated that cells should be seeded at a low density of 8,000 cells per well in a 96 well plate. 48 hours should be used for incubation time to allow for complete growth. 2 mg per mil of collagen concentration was determined to be optimal as it allowed for the collagen gels to maintain their structure throughout the growth and imaging period. This experiment also indicated that very little motility is seen in these cells at baseline over the 8-hour imaging period. Future directions for experimentation include that analysis of single-cell spatial movements to determine baseline characteristics. Possible perturbations from various drugs can induce motility response and will be used in the future to speed up movement and allow for deeper analysis. A big thank you to the Philip Lab for making this research possible, and a special thanks to Dr. Jude Phillip, Anshika Agrawal, Pratik Kamat, and Teja Stewart.